tox as an example of a botulinum medicine. So just a bit of background about the problem. Well, essentially, when you make these biologicals, clostridial medicines, they have to undergo rigorous safety testing. So animal use is extensive in the production, and I'll focus on tetanus vaccine for this talk, but it applies to botulinum as well. So tetanus vaccine is an essential global medicine with some 84% of the world's children immunized with at least 3 doses of the vaccine. And during the production process, many animals are used. So essentially what happens is that in order to make the vaccine, you take the bacteria that ultimately produce a toxin, tetanus toxin, that's the pathogen causes the disease, tetanus. So you make the bacteria in a big, large-scale fermentation, they produce this toxin, it gets purified out, and then use chemical, usually formaldehyde, to deactivate it to become tetanus toxoid. And that's what goes in the jab. So before you put it in the jab and give it to all the world's children, you have to make sure that there's no residual tetanus toxin left inside. And that's where animal testing comes in. And unfortunately, it could be some of the most severe animal testing on the planet. It often has a lethal endpoint, and that lethal endpoint is often death by suffocation, respiratory depression. It's very expensive, requiring lots of infrastructure and skilled personnel, including in-house veterinarians. And generally, it's done at the production site that's really built into the production process. It's a very lengthy.
I've got to pick up. You're missing. The kid's going to be waiting.
It's not my one.
Or is it not?
I hope. So anyway, it's at least 3 weeks, the test, and animals die for other reasons, it's death readout essentially, and because of that, when they die, then you need to repeat the test, it's very inaccurate, delays to market are caused. So basically, so we collaborate with the UK MHRA, the UK regulator. They actually brought this problem to us. So as experts in sort of the underlying biology of clostridial toxins, they came to us about this and asked us to develop technology that had to satisfy a certain set of criteria to essentially create a new global gold standard to replace this animal testing. And that's what we've done. So we developed the technology and packaged it into a sort of a user-friendly cell-based assay using pretty kind of crude cells that you render within a couple of days to become more nerve cell-like, which are the cells where these toxins act. And then it's all packaged into this thing called ELISA. It's animal suffering-free, and it can reduce their QC costs extensively. It's a 6-day technology instead of a weeks to month test. So it can solve this sort of bottleneck in production pipeline. It's very, very specific on like the death readout, and ultimately it can reduce vaccine shortages, which can be a problem.
Okay, I mean, just on that note, can you explain how much it would cost someone, or how many animals are used in the testing process normally, and the potential cost saving and time saving of that? Yeah,
I mean, well, it varies, you know, from manufacturer to manufacturer. So they produce on massively different scales. And just an example for numbers, I have to my head for a botulinum manufacturer that I'll talk to you about. They're using 80,000 animals per year for their.
80,000.
Yeah, and they run out pretty quickly as well. So they've already run out this year. We have to go up to get through the rest of the year to test. So on the tetanus side, they use fewer animals, but this lengthy test, this is a real bottleneck for them. So you could imagine going from weeks to months to a 6-day test. You know.
And with the new sort of guidelines from the FDA on moving towards animal-free type testing, how has that impacted your business? How lucky are we to invest in you just before that?
Absolutely. I mean, the time is right. The time is absolutely right for this. So everybody wants, you know, animal replacement tech at the moment. So this is perfect. And it's here, you know, it's working really nicely. So yeah, it's the perfect timing.
So obviously you knew it's coming, you know.
Is there any threshold for the quantities you need to detect?
Absolutely.
To the sensitivity of the assay?
There is, yeah. So this is sort of a key one of the key things that we had to make a highly sensitive assay. So these toxins are the most potent on the planet. So it has to be, so it's within what you call like the femtomolar range. So really, really tiny amounts we need to detect. And, you know, we didn't do it straight away. It took us several years, to develop this.
I imagine these antibodies are commercially available.
The antibodies we've developed ourselves. So some companies have tried to develop it. Some of the bigger companies, some of the regulators, they haven't managed to. They've made polyclonal versions. So we've made a fully recombinant monoclonal panel of antibodies that nobody else has managed to do. And actually we just filed a PCT on those particular antibodies just earlier, or at the end of 2024. Yeah, so that's one of the key things we have, as I say here. For a global gold standard, you sort of need to satisfy several criteria. You've got to cover all the physiological steps of tetanus intoxication. We have a competitor. It's the binnacle assay developed by the Polar Ehrlich Institute in Germany, but it's basically, it's probably not suitable for a global gold standard. It could be used within Europe. But if you want to sort of a global gold standard, you're going to have to go with a cell-based technology. So theirs isn't cell-based. It doesn't cover all of the physiological steps, only some, and that can cause problems. So ours is this sort of cell-based assay. And their tech is also, they have a polyclonal antibody. They've had issues with reproducing another batch of the polyclonal antibody. Whereas as I just asked, we've got a reproducible, scalable monoclonal that sort of underpins the tech. And then, for this reason, the MHRA have told us that, you have to have these first two sorted before you can really satisfy more conservative regulators globally. So the binnacle, the bilateral will be it's going to be accepted in Europe, but even in the US, they're not. It has to be fully physiological, and like the other places in the world. So we satisfy that. So we know in terms of sensitivity, it performs in-house, and actually we've translated it to the MHRA for both botulinum and tetanus at this point, and it's performing nicely there as well. And now we need to test with real-world commercial vaccines. And this is, I'll tell you about that in just a second, but so far we've had really great positive sort of results from a botulinum manufacturer and a veterinary tetanus manufacturer as well. So we currently have an international consortium study to evaluate the technology using real-world commercial materials. There should be a phase 2 here. We're just finishing up phase 1, where we've recruited major vaccine manufacturers into our study. And this is coordinated through the UK MHRA. And then we test their materials within our tech at Sheffield. It's anonymized to us at this point. It's gone really well. We're about to finish up in the next couple of months on that side and go into phase 2. And this is where we'll actually translate the tech over to the end users, the manufacturers. They will perform sort of a very small in-house evaluation where they have a prescribed set of experiments to do with our tech and then report back to the MHRA. And the whole idea behind it is to sort of get a collective industry-wide sort of push to overcome the regulatory hurdle here. And in terms of market traction, in our study, we've had amazing uptake. So we estimate that the manufacturers involved collectively make more than 90% of the world's tetanus vaccine. And that's on the human side. We've also allowed some veterinary manufacturers, some major veterinary manufacturers in as well. Yeah. So in terms of the market, we estimate that it's worth 50 million per annum. We're currently focused on tetanus and botulinum B. This is what the tech as it stands is immediately applicable for. We're also developing for botulinum A and then other clostridial vaccines as well. And in terms of the business model, we will do it via contracts for access to the technology and also our expert technical support, and then also supply of the critical reagents. And MHRA and the EDQM are interested, have asked about housing our antibodies and cells as reference reagents, and we'll talk more to the companies about how they want it delivered as the project goes on. And in terms of market traction, as Sunil alluded to, I was in Washington earlier this month negotiating a project with a botulinum B producer. So they've already had a trial with us for the tech, and they now wish to implement the technology with their CRO as their main batch release potency and stability test for release of their product, which is available in the US market. And they want to do this by Q1 of 2027. As I said, they use 80,000 mice per year to do this. And we're just literally, we've just finalized a development project with them, which will run for the next 18 months. And next week we've agreed to finalize the sort of the licensing. So it's, you know, in excess of $1 million per year in licensing fees alone. So we don't have to do any testing ourselves. We're giving them access to the tech for that, and their CRO will carry it out. So it's a really nice model for us. And as Sunil said, we've just closed seed round for 300K. Sansanima has a post-seed valuation of 3.3 million. So yeah, you can get my contact details if you want to talk to me a bit more about it. But yeah,
Thank you so much.
Thanks.
Two questions actually. The first one is, why does it take 6 days to do your ELISA assay? And the second one is your PCT covering the functionality of those antibodies or the actual composition of matter of those antibodies?
So the assay takes 6 days because you have to render the cells fully sensitive to the toxin, and that requires a chemical differentiation. Yeah. So the neuroblastoma, there's a chemical differentiation rendering them fully sensitive. They're a reporter cell line, and they've been engineered to be highly sensitive as well. And they have sort of a reporter molecule in there which allows us to do luminescence-based detection. So that takes 3 days, and then you apply the toxin for 3 days, and on the last day you run the ELISA. Yeah. And then we filed for, I'm sorry, I've got a cold, so I'm just starting to cough in here today. This is my main fear. So basically we will, we filed the antibody. You asked me, was it, so yeah,
Was it the functionality of those antibodies, those recombinant antibodies you made, which was the really smart idea?
Yeah.
Was it the functionality of those, or was it the actual nature of the antibodies themselves, the sequence of each of those antibodies?
We've patented the sequence of the antibodies, but we also have another patent as well, the underlying tech, which is basically that they're sort of a reporter cell line that carry the final target. And normally in the body, this target, once it's attacked by tetanus or botulinum toxins, it's rapidly broken down, and it evades sort of an immunological detection with like classical methods. But we've stabilized it so it hangs about, and you can readily detect it with antibodies that are specific. They only see this target if it's been cut up by tetanus toxin or botulinum toxin.
Matt, and then we're going to have to call it Matt.
Where do you see payor uptake here and revenue growth? And then what's your exit strategy? Are you going to partner with strategic like Vestas, or are you going to be in embedded government contracts? You know?
So we're leaving it open, but like spoken to some of the big CROs who are, you know, at the moment, some of them do animal testing on behalf of some manufacturers. Some keep it in-house, and some outsource it. And they're definitely very interested in taking this.
It's further down the value chain. Though.
Yeah.
Big deal.
I think it's evolving. I think part of the reason that Sheffield Angels and Andy and I came on the board is to provide some support on how to sort of really sort of monetize the platform and what Katie said. I think it's evolving. Is that fair?
Yeah, I think that's fair. I mean, it's definitely worth a lot of money to a CRO. They would the go-to CRO for manufacturers, you know, with this tech. So, but you know, the good thing is that, you know, we don't have to do. Our business is the development of the technologies. So it's a licensing model. So we don't have big overheads in terms of having to run any tests and things like that. So, you know, within the next year, we should have annual licensing revenue in excess of £1 million, hopefully.
Thank you for coming all the way from Sheffield. You know you're not well.